1 action. I'm just going to test out the chat before we begin. It works definitely perfectly. Let me check something out as well. All right, perfect, ladies and gentlemen. We can begin in 3, 2, 1, action. And this is going to be a different kind of stream. It won't be a gaming stream. It's going to be more of a scientific stream. Today, we're going to be taking a look at an animal cell versus a plant cell. Again, I know it's not gaming content, but I thought it would be interesting to display and showcase a little bit of biology on Twitch and on all of social media platforms, on YouTube and on Facebook, because this is going to be going everywhere. So we are good to go, and we are ready to go. Actually, let me just set something up. Perfect. That's a little bit perfect, and let's begin actually with a little bit of context. And just to give you a reason why we're doing this, I think it would be interesting to know because the thing about cells is that they come from a very large domain. Cells come from the domain, I believe it's domain, reign, family. So I believe it's definitely domain. But anyways, cells. Human cells in this context, so our cells, what our body is made out of, comes from the domain eukarya, meaning that we are eukaryotic. And what does that mean, eukaryotic? As you can see, this is a fact. Eukaryotic means that you are that it means real nucleus. It means that the cell has a real nucleus, and we're going to see that why in the actual microscopy that we're going to be doing a lot later. Now, the reason why we're comparing a human cell to a plant cell, you could ask yourself, well, there's actually absolutely no context to it. A plant is a plant, and an animal cell is an animal cell. There's no correlation together, but again, the fact that both of these cells come from the same domain, meaning from the same family. You can think of it as coming in from the same family. If you're new if you're new into biology, you could think of it as the same family. And we're going to see why is because these cells have a real nucleus, as you saw in the previous image. If I can hover my mouse over here, this is a human epithelial cell, which we're going to be looking at, which, as you can see, has a nucleus over here. It's not a stain; it's a nucleus. And as you can see, there's a nucleus over here inside of the plant cell. So that's a little bit amazing. In terms of similarities. Now, here's where the differences arrive. The human cells, in this case the animal cells, but the human epithelial cell that I just showed you previously, are heterotrophs. What does that mean? That's a fancy word for saying that they need to consume organic compounds, well, I would assume that they're organic chemicals. So anything from like food, anything from like anything that is made out of anything that is organically chemical, that is digestible, that is heterotroph stands for in a nutshell, meaning that you hunt for food, you need to gather resources. On the other hand, plant cells are autotrophs. What does that mean? That's another fancy word. There's the word troph inside, so you know that those words are kind of similar, but very different. Autotroph means, in a nutshell, that you make your own food. You're able to make your own food, which is completely amazing. I mean, shopping, grocery shopping would be completely gone out of business if humans could make their own food, you know? But with that being said, if we look at an animal cell under a normal scientific microscope, which we're excluding photonic ones because we're not there yet, this is more of amateur microscopy, Identifiable parts of the cell are the nucleus, as I've just seen, the cytosol. And what is the cytosol? Well, the cytosol is, I did an excellent video explaining mitosis, and I explained parts of the cell inside of that. The cytosol is essentially the liquid that is inside of the cell. So, if you've done normal biology or normal science in school, many of your teachers would have told you that it's the cytoplasm. Well, that's not entirely true, because the cytoplasm is not the liquid itself. The cytoplasm also contains the organelles. The organelles, which are the organs, in a nutshell, they're the organs that the cell needs in order to function. So in order to survive, in order to keep living. And so the cytosol includes the organs and the liquid that stays inside of it. But the liquid itself is called the cytosol. So now you know what a cytosol is. And finally, a cell membrane. Now, the cell membrane is what goes around. It's essentially what gives the cell its shape. Because without a cell membrane surrounding the cell, everything would just splooge out, right? A cell wouldn't be a cell anymore, and the organs would be free to roam around, and then you wouldn't have a cell anymore. So all of these organs, all of these which are organelles, the actual term is organelles. I'm pretty sure I'm making a lot of scientists and a lot of biologists very angry, and they're probably like, stop saying organs, they're not organs, they're organelles. But it's to basically make amateur people who want to get more into biology to make them understand what the concepts are. Anyways, back to the cell membrane. So the cell membrane is what keeps everything inside, and that is made out of a phospholipid bilayer. I'll get back more into that once we take a look at our human cell that we have, an actual human cell that we're going to take a look at under the microscope. But moving on to the plant cell. So again, excluding photonic microscope, identifiable parts of the cell are again the nucleus. As you can see, there's a similarity, cytosol, and a cell wall. This is a big difference over here. There's a cell wall and not a membrane. Actually, they do have a cell membrane. Plants do have a cell membrane, but they're mostly surrounded by a cell wall. And this is where we actually go into the big differences. Which brings me to my hypothesis. So, compared to a human cell, a plant cell will have the exact organelles as the human ones, except for the additions of plasts, or chloroplasts in this case, vacuoles, and a cell wall. Now, I didn't talk about the chloroplasts. The chloroplasts are what makes a plant, making it autotrophic, meaning that it makes its own food. How does it make its own food? Well, from solar energy. Heat from the sun. Heat from the sun goes, and we all learned photosynthesis, how it works. Sun comes down onto the plant. Plant absorbs the heat, making it as such that the plant will be able to make its own food. Moving on to vacuoles. What are vacuoles? Vacuoles are essentially they're like this big pouch that, because let me put it this way. A plant cell specifically, will have like this pouch inside of it where it will store all of its organic compound, all of its liquid, and it's going to store as much as it can. Generally, all I know, and correct me if I'm wrong, definitely try to correct me in the comments and everything like that. But generally, it's going to store liquid in that vacuole because it wants to have as much liquid as possible to be what we call turgescent, meaning that it's accumulated a lot of water inside of it. and it wants to be like really, really, really big in size. You know, it doesn't want to be in a, I'm not going to say hypotonic or hypertonic environment. I'm looking for a specific word that's like in between turgescent and a flaccid cell. I'm not sure what the word is, but there's a specific word for that. It doesn't want to be a stable cell where it's like a normal size. No, it wants to have as much big of a size as possible. It wants to like expand, but not explode at the same time. And that's what's kind of like the cell walls are kind of there for as well, is to keep it from kind of like just exploding. And the vacuole is also really good for that, because the more water it takes in, it just doesn't explode in the end. And then finally we move on to the cell wall. The cell wall, like I've explained, it's a wall that surrounds the cell, that surrounds the membrane, and making sure that the cell remains in place. And we're going to see that, like I said, I showed it, you saw it a little bit on of the first page of the presentation, but I'll show it later on if I can with my mouse, because I am currently on a computer right now. So moving on to a bigger picture on the organelles, because like I said, the organelles are really important for the cell. It's what makes the cell live. It essentially ensures that the cell keeps on living. It keeps on keeping on, you know. But yeah, in a nutshell, organelles are the organs which ensure living conditions for the cell, which is like what I've already said. Organelles are made up of molecules. Structurally, they're made up of different type of organic molecules. For example, I'm going to hover my mouse over here, right over here, this thing over here, and then this thing over here, which is the phospholipid bilayer. It has the word phospho, meaning that it has phosphorus, where we find phosphorus. If I'm not mistaken, it also has, it's a phospholipid bilayer, so it has lipids, made out of lipids. So if I'm not mistaken, I wish I had my, actually, I'm just going to open it, my textbook's over here, because it has a tail, if I remember correctly. Let me open my biochemistry here and go into lipids, and then go to the section which says it's not made out of triglyceride, it's made out of I'm sorry, I'm going to screw up this word so badly. They're made up of phosphatidylcholine. Phosphatidylcholine. Anyways, that's a fancy word just to say that the head is hydrophilic, meaning that it loves water. And the tail, and I wish I could show this on screen, but once I edit out the video, I'm probably gonna show this on screen as well. But a hydrophobic tail. And the hydrophobic tail is made out of those fatty acids. Those are actually called lipids. Lipids in French, fatty acids in English. That was the word that I was looking for. I'm kind of a little bit bilingual here, but it doesn't really like matter or anything like that. Actually, I guess it does matter if you're trying to learn as you tag along. But anyways, like I said, made out of molecules and organic molecules, actually. And each organelle will have a very specific function to accomplish. For example, if we take over here the mitochondria, as we know and as we have been taught, and it's like the most very basic thing of what makes energy. We've all learned it. It is the powerhouse of the cell. Why? Because it makes energy. It makes adenosine triphosphate, ATP. Yes, that was actually what I was thinking about. For example, lysosome. If I'm not mistaken, the lysosome has the ability to disinfect the cell from any nasty chemical compounds, or to digest, if I'm not mistaken. It would be to digest. We have the endoplasmic reticulum, and here they didn't actually precise it, but there's two of them. There's the smooth one, and then there's the rugged one. But here they just placed the endoplasmic. This isn't a very accurate image. I don't even know why I placed this. But anyways, cell membrane. Oh, they actually did showcase the cell membrane, which is, as you can see, for the plant cell, it actually does have a cell membrane. But it has an extra layer, which is the cell wall. It has chloroplast, it has a vacuoli, and as you can see, we kind of find the same exact organelles in this type of cell, in both cells, in the plant cell and the animal cell. Cytoskeleton, cytosol, nucleus, peroxisomes. Oh, the peroxisomes are actually the one that disinfects. Sorry about that. The lysosome is actually the one that digests. Sorry about that. Golgi apparatus, and etc. So each one organelles have a specific function to accomplish, and that's what makes the cell a living organism. That's what makes both cells a living organism. So finally, moving on, a few references. As you saw at the beginning, I'd like to thank Dr. Shikajagi for his human epithelial buccal cell that was found on the internet, that was accessed through metadata. Thank you to Jacob Straka for his animal and plant cell image that he did, which was the graph that you saw on the previous slide. And then finally, an excellent reference I highly recommend reading is the Campbell biology book. This one is specifically taken out from the French edition, but anyways, I digress. Excellent reference to go and check out. Now, I'm just going to change a little bit the screen a little bit so that we can prepare. And the first thing we're going to look at is going to be the epithelial buccal cell that was sampled from an actual human being. And that human being is actually myself. If I can just get my... Oh, sorry, I was a little bit too high. I was defaulting my... But anyways, you know what? I'm just gonna let you guys see what I'm doing. I'm basically trying to verify my my camera if it is working. Okay, so it is working. I do have image, but it's just that I don't have, I just don't really have anything to look at, which is weird. Oh, there we go. There's images. Perfect. All right, we got, I'll probably set the, yeah. So the light is basically at 1. We got basic light, and I think I'm going to change it to a different lens. All right, we've got perfect, and we're just gonna try to look and see if we can spot an epithelial buccal cell, courtesy of myself. I don't think that's an epithelial buccal cell. I do not think so. Give me a second. Actually, it might be one. Let me just change it. Change from 4x to 10x. Change the light. Make it a little bit brighter. I still don't think that's a cell. Let me just center it. It's not a cell because what I'm trying to look for is nucleuses, is a nucleus, or nuclei, nuclei's. nuclei. If I'm trying to spot them and I don't see them, that's not a cell. So let's go back to our 4x, and it takes a little while until you kind of just like spot them or something like that. It takes a while. There was a bunch of them that I found. I'm trying to find like the ones that are like grouped up, that are like just kind of bunched up a bit. There were some of them that I kindly saw, and I think these are dust particles. Yeah, these aren't the this isn't a cell. So like I said, it takes a while to find, but once you do find it, it's definitely something worth looking at. And oh, I think, yeah, these are them. These are the cells. Just gonna see if there's like the bunched up the bunched up kind. But yeah, these are the cells. I'm just gonna go back to where I was if I can. And there we go. There we go. That's actually a perfect one that I just spotted. I'm just gonna try to center it. I'm just gonna try to center it right here. We're trying to do some cell hunting today. And I found my target. All right. So as you can see, this right here is our epithelial buccal cells. Interesting fact about the epithelial buccal cells. The word epithelial means, well, it comes from the word epithelium, meaning skin. Buccal, meaning found in the mouth. And now I'm just going to put my 10x lens. I'm going to up the... I also apologize if there's like any like seizure. I'm trying to go as slowly as possible to not cause a seizure. And did I lose it? I think I lost it. I think I lost my cell. And these aren't the oh, there's one actually, but it's like on top of the everything. The dust and the water is on top of it, so you can't really see it really well. It's like right here. It's like right here. But yeah. And so let's, oh, I think I found one. There we go. This was I think this was my target. I think this was my target. Yeah. And as you can see, as I've stated, the identifiable, I'm trying to like center it perfectly. I think it's centered enough. All right. I think it is perfectly centered enough. But, like I said, as you can see, the identifiable parts of the cell is its membrane, which gives it its shape. As you can see, look at the specific shape it has. And the nucleus, and the cytosol, which is everything inside of here. The organelles are unfortunately way too small, and they require a photonic microscope. Before I change the lens to a 40x one, let me just show you guys what the different what the different lenses are actually look like. This is I think this is a normal one. This is I think I'm at C. I think I'm at colorless right now. This one is at R, which I think is red, a red lens, but it's not really doing much because I think that the on a white balance. I'm trying to like change the white balance. Again, I'm terribly sorry if people have seizures. Like I said, I'm trying to go as slowly as possible to probably accommodate for the people that do have seizures. Hue. Can I change the hue? Can I change the saturation? Nope. I'm just going to go back to default. Oh, I'm just going to go back to default. All right. So you can't really like see the different lenses that I have, which is kind of what sucks. Normally what you would do with this cell, you would color it, but I unfortunately don't have methylene blue or red in order to create the proper chemical reaction to it's a small liquid that you get, methylene blue or methylene red, in order to color the cell and to make it able so that you could see it. But anyways, I digress. Let's move on and change the lens all the way to, I'm just going to up the color. I'm going to change it back to, oh, there we go. I think we can see something. And this one is at colorless lens. And, oh, is this one? Is this, I think I'm going too fast. Screw my, okay, there we go. It's at least centered enough. Is this one? No, it's a dust particle. It's a dust particle. That's so effed up. So yeah, I lost my cell with the 10x. Actually, it's 40x. You just have to like look around. Oh, is this one? Is this one? Oh, yeah, perfect. Perfect. I think I got one. Even though it's like super dark. Even though if it's like super dark, I don't think, I probably still think that it's a, I don't think it's a buccal cell, because the nucleus looks too effed up. Look at what I'm looking at here. This is supposed to be the nucleus, which is kind of screwed up. It doesn't have like the actual nucleus shape. Correct me if I'm wrong. If anybody else is like a better observer than I am and has done like and is a veteran at microscopy, let me know if that is an actual cell that I found, because I think that's like the closest one that I could find. Yeah, I think those are the closest one that I could find. I'm gonna try to see if I can find any. Oh no, that's dust again. Dust again. I don't think we're gonna find any. Not at 40x. Not at 40x. I have to go back. I think I lost my cell. Either way, I lost my cell. So careful for the light. And let's try to see if we can find other buccal cells. Oh, and I think I found one. Oh, no. That's water. Or maybe that's one over there. No. That's interesting. All right, let's see. And I think that this is like the tip of the water. I think this is like the tip of the water. Tip of the water. I'm gonna put the exposure a little bit down a little bit. No, those aren't buccal cells. Is this one? No, those are still like water particles. Water particles, a bunch of water particles. Buccal cell. Where are you guys? Where are you guys? See, if this was, well, I mean, you could argue that I should have placed methylene blue, but it wouldn't have made a difference because, like, it wouldn't really have made a difference. Because, yeah, it's useful to like know where to spot it on the microscope. But the thing is that the buccal cells are actually transparent. If I can't find them in the next 5 minutes, we're probably, oh, and I'm like at the tip of the, oh, actually I wasn't even looking at the right place. I wasn't even looking at the right place. Nope, still nothing. Still nothing. I had so many of them. I spotted like 2 or 3. No, these aren't our buccal cells. Yeah, I think I lost them. I lost them all. And the worst part is that I didn't even like Q-tip myself. Well, I did Q-tip myself. I Q-tipped the inside of my mouth, but so like I said, I Q-tipped like the, oh, is that one? I think I found one. Bingo. Bingo. Bingo right there. And I think there's like, okay, there's like a few, there's like 2 more. There's like one over here, and then one over here. And let me just put the contrast like real low. Oh, actually, we're going to swap to the 10x. And I think I'm just like pinpoint. I just, yeah, perfect. Perfect. Bingo. I'm like perfectly on it. Bingo. Sometimes you just got to be patient with these kind of stuff when you're doing microscopy and trying to look at stuff under a microscope. So the big question is like, why buccal cells? Well, they're like the most basic thing that you could take a look at, and they're like the most like basic basic organism, basic living organism. Of course, these are dead because they're not moving. But then again, like cells take their time to move. They move at like, I don't know, per hour. You could like see movement if you just fast forward the camera a little bit, but that's as much as it is. But these ones are dead because you can easily like pick them up. And what happens with cells is that they want to take as much space as possible, but when they die, that's when new ones grow. And I did an excellent video on cell division. If anybody is interested in seeing that, it is available on YouTube and on my Facebook page. If anybody is interested in seeing that, definitely have a look at that. It is definitely worth it. All right, moment of truth. Let us see. I'm just gonna bump it up a little bit to the max, and I'm going to see if I can put the 1000x. No, I think it's the same problem. It's really hard to like pinpoint a cell with this thing because it's super. I'm just gonna bump up the exposure so that people can see what I'm doing. The reason being is that this lens, not lens, but optic, I'm not sure what it's called, but anyways, this like 40x lens. Ah, there we go. I think it's spot one. Spotted one. Oh, I lost it. I didn't lose it. Can I center it? I'm going to try to center it. I'm going to still trying to center it as much as I can. I think I'm good like that. The issue with this, the issue with this is that there's two cells on top of each other. The nucleus is right here, but it's just that they're overlapped on top of each other, and it doesn't help because you have dust particles, which are like water particles that I wiped off from this. Can I center it? Oh, I managed to center it. Perfect. Can I focus on it? I can focus like a little bit, and I think you can see two distinct nucleuses or nuclei. One over here and then one over here. And so, as I was saying, what's really interesting about buccal cells is that they're like the most living basic organism of your body to look at. You can easily like just view them under the microscope, and it's so interesting because your body is made up of these tons of cells. Granted, they're not the same cells because cells, each one of them has a specific function, and the way that works is that each cell that has a specific let me start from the beginning. If a cell performs, just know this, if a cell performs the same function over and over again, and you have multiple cells doing the same function over and over again, then that's called a tissue. And tissues that perform different functions altogether form an organ. And that's true because we see that in the function of, for example, let's take the skin. The skin, which is the largest organ inside of the human body. You have the dermis, epidermis, and etc., right? Now, you have different cells at different layers that perform different function, but they're essentially part of the same organs. Again, cells, tissues, organs. And from organs, you drop down to tissues, and then you drop down to organs. That's a little bit of the cell biology structure in a nutshell, if you can at least remember those. If I can name you all of them, I'll probably sound too overpowered, but that's pretty much it. There's also organelles, because you start from the smallest, which are molecules, then you move on to organelles, and then the cell, and then etc. And I think there's like 9 or like 10 stages, but whatever. So that was it for our epithelial buccal cell. I will now go back to ensure that I don't seizure anybody, because again, it's like very I don't want to seizure anybody, because we're manipulating like really small objects and with light and everything, and it can be strobing at times. I'm looking at it on the screen and like swapping like really quickly to like white or to like a white screen. I heard that it can cause issues. With that being said, let me default it back and then change the light. And we are good to go, ladies and gentlemen. All right, so what we're looking at here is our plant cell. In this case, I have chosen an onion. The reason for that is because they're very easy to look at, and they're like really easy to strip off, essentially. So what we're looking at is like tons of them. As you can see, there's like tons of cells bunched up together. I just centered one of them right now, and it's, if I can put my mouse and hover, it's right over here. This one in the middle right here. That's a cell. Why? because you can easily recognize it from the cell wall. From the cell wall, and that's a huge difference between a animal cell and a plant cell. Now, I don't really see a nucleus here. I'm currently at 4x of optics or whatever it's called. Let me know in the comments what it's actually called, because I call it lenses, optic lenses. But with that being said, can I lower the light a little bit? Can I up the light? Okay, I can do that. So that's good. And as you can see, they're just kind of like bunched up on top of the other. You can see right below there's other cells, unless that's dust. There's a cell over here, there's a cell over here, and there's like one. I'm trying to like do their shape and everything like that. But anyways, now that that's done, the only unfortunate thing, I'm going to try to see if I can like switch lenses over here. Oh, I swapped it to green, I think. I swapped it to green. It's really hard to like change the apertures to like different colors and everything, because it kind of like makes the microscope move as well. The microscope isn't stable, but it is what it is. With that being said, let's swap lenses. Let's go from the 4x zoom to the 10x zoom. As you can see, everything is blurry, and it looks like really trippy. This looks incredibly trippy right now. I'm looking at a bunch of stuff, but not really. Oh, here's a few cells that you can make out. And if I'm not mistaken, this is either dust, water, or it's actually a nucleus. But with that being said, I think what you can do is I'll try to see if I can focus in. No, I think that's the max that I can focus. Let me see if I can find, because like I said, they're all bunched up together and it's not very good. Ooh, perfect. Oh my goodness, these are perfect ones. There's two perfect ones that I just found. No way. Nice one. All right, so as you can see, I'm drawing the shape right now with my mouse. Right here, as you can see, the cell wall, and there's right one next to it, right? Right? Is the one that you can make out. Awesome, right? And let me see if I can like put the contrast. You can see them even better now. Oh my goodness. No way. This is amazing. And as you can see, this is right here when I'm like circling, hovering over, is the nucleus. Nucleus right here, nucleus right here. Now, as I said, there's an unfortunate thing that I can't do, is that I can't put like the 40x. I can't put that one in because, well, it goes way too far. It's way too big for it. It's not like we're trying to spot organelles. It's not like we're trying to like definitely spot organelles, like I said. But what happens is that it'll be way too big. It's going to be like this over here. It's going to see this section and we're not going to see anything. We could probably make out the cell wall, but you can make it out from here, which is definitely perfect. It's definitely a perfect view. With that being said, as you saw in the PowerPoint presentation, in the slides presentation, what happened was that I was speaking about the cell wall, and I said I was going to come back to it. Now, the cell wall compared to the cell membrane, as I've said, this is to ensure, actually the cell wall has multiple functions that we're going to get into, but it's one of the primary functions is to ensure that the cell takes its adequate amount of space that it can take. As you can see, this is like the space that it can take, right? Another function of the cell wall is something called a plasmodesma. Now, that's another biological fancy word that is just to say that the cell is able to share structures between it. Well, structures, what am I saying? Organic compounds between it very easily. So if one of them is missing a certain organic meal, if I can say it that way. I don't have the correct word for it or the right thought for it. I can't really think of it right now. But anyways, they share. It's like small gaps that they use. The plasmodesma is like small holes that are connected between the between the cell walls that are made in order for the cells to pass food around or energy sources. I've also seen, I don't know if you guys know this guy, but he's like really super good with microscopy. He used to be a microbiologist, or at least he's still a microbiologist. He's a microbe hunter. So microbe hunter, really recommend checking out. He doesn't do live streams though. He just does videos though, but he's really good at them. He's really good at making microscopy videos, and he's a veteran in the game, and I commend him for that. But yeah, let me see if I can change the lens to a more colored one. This one's more darker, so I need to like bump up the light. Maybe less light. But yeah, this is the best that I can do for like putting it in another color. And definitely this is like I said, this is the best I can do for putting it in a different color. I thought I was actually gonna need to use my my epidermis onion slide, pre-made slide from actual microscopy biologists. The reason for that is that before starting the stream, I was trying to get nuclei, and I couldn't really find any of them. Like if I change, I see that this one has a nucleus. This one doesn't have a nucleus, but I'm pretty sure, yeah, you can barely see it. You can like barely make it out. And I was like, oh my goodness, this one has a nucleus. Oh, I really picked a really good spot to like view. This one has a nucleus. Yeah, so I really picked a really good spot. Oh, do these have a nucleus? Yeah, these ones have a nucleus. And like I said, I really picked a really good spot for to view these kinds of things. With that being said, let me know in the comments what y'all think about which one is your favorite. I'm curious to know. Did you enjoy looking more at the buccal epithelial cell, or was it more enjoyable to see the to take a look at the onion cell, which is also a plant cell? So in general, most plant cells will have the same structure. You're gonna have the same structure. So a cell wall, cell membrane, nucleus that you can identify under a microscope, and that you'll be able to check out and see what it actually looks like. All right. With that being said, I think I've done most of the. I wish I had more people watching the stream and everything like that, but Yeah, so I wish I had more people watching the stream like that so that they could ask questions. Maybe I could like answer them and everything like that. But again, I'm not like a biochemist or anything. If you're curious, like I can still put the 40x lens. That lens is like super huge, but first of all, you won't see anything. I have to like bump up the light to like the maximum, and then I have to bump up the exposure really, really high. And as you can see, you can't like really see much. And if I just move it one bit, and this is also trippy at the same time. But yeah, as you can see, you can only make out the cell wall, which is like super weird. So that's it. You can just make out the cell wall, which is like WTF. Can I find a nucleus to like pinpoint what it looks like? I don't think I can. So this is what the 40x looks like, which is you can't really see anything. So back to the 10x, we're going to go back, and sorry if this caused probably seizure stuff. Like I said, I'm trying to like limit the amount of seizures. I think I lost it, but you can still make out the nucleuses. Oh, this is water. This is water, but you can still like make out the nucleuses inside of them. And I'm back to the oh. Can I actually? Oh, perfect. All right. And we're just strolling around, and it's quite interesting to know what these things are made out of. It's definitely interesting to know what really gets me going is that to know that somewhere, somehow, millions of years ago, we had that last known common ancestor, which derived from eukaryotic, again, eukaryotic meaning that there is a real nucleus, knowing that there was a last common known ancestor that was eukaryotic and then diverged. So it's interesting to know that we're all part of like different cells, but still, it's interesting to know that these cells evolved a certain way, and these cells evolved a certain way, and other cells evolved to do other certain things. So it's quite interesting to know that. And I'll just like leave it here just to showcase. To me personally, what I enjoy most looking at was perhaps the buccal cell. And there's not a lot of time left, so what I'm going to do is that I am going to stop that sharing, and I am going to go back, and we're going to take a look at the buccal cell. These are pre-prepared slides, by the way, that I've prepared, that I ensured to prepare in order to view and to see, actually. Let's put it back, and let's go back to some buccal cell hunting. All right. Let's see if we can find anything before like time comes up. There's like 8 more minutes left on the stream, but I'm gonna use that time like really, really wisely in order to like view as many cells that I can. And if you're wondering if this is one, I'm gonna center it. I'm gonna try to center it. If this is one, I don't think so, because it mostly looks like a water particle. But yeah, I'm trying to find. Oh, I think I found one. Oh, no, that's not one. That is not one. This microscope that I got was really good. And if you'd like to know if I'm like stepping away from gaming or anything like that, not really. It's just that I gotten as a respiratory therapist. One thing that I do is that I love doing research. And the first thing that I did when I got this microscope was like, okay, let's try to see if we can live stream with it and try to see like cool things. And the first thing that I wanted to do was to do like the buccal cell thing, comparing it to animal cells, comparing animal cells to plant cells, because they're like the most basic thing that you can do. And I don't think that we're going to see any more buccal cells. I'm trying to hunt here kind of like last minute. I'm trying to hunt for if I can spot a buccal cell. There's like 5 more minutes left to the stream. I don't think I will. But interesting facts, if you are interested of learning more about cells and more specifically how they divide, I would definitely tell you to consult oh, is that one over there? I think it is. I think it is. It's small, but I think it definitely is. I can't seem to put the lens. Oh, okay. I managed to place the lens. I'm just gonna up the light a little bit, and just gonna go up. Oh, it can't go up anymore. Oh, I lost it. That's sad. That sucks. I kind of lost it. Yeah. I'm sad now. I think that was one, but whatever. With that being said, the stream is coming to an end, and I would definitely like to take this time to thank anybody or everybody who kind of takes the time to, or who are interested in science and wants to take the time out of their day to like kind of learn more about cell structure and how cells are made out of, and the interesting parts of the cell, and etc., and all of that fun stuff. Really, it's what gets me going is like that this is made up. This is definitely, and the functions of it, the functions of the cell, what they do in our body, the different functions that they have, is definitely like very, very interesting. But I don't think that in our last... Oh, I think I found one. Yeah, this is one. Yeah, that's one. That's one over there. All right, perfect. I think I need to boost it a little bit. No, I think I need to go a little bit up. There we go. There we go. Is this... Huh, there's 2 nucleuses. There's 2 nuclei, if you've noticed. No way. Is this what I think it is? Is this a cell during cell division? That's so interesting. Oh, no, I think that there are 2 cells on top of each other, and they just happen to... Yeah, no, I think there are just 2 of them on top of each other. Okay, no. That would have been really super cool, though. It's like seeing mitosis actually happen here on this channel. It would have been super cool. But anyways, we ran out of time. And like I said, I'd like to thank everybody who tuned in or who will be tuning in for future reference, and that have tuned in. Thank you so much for watching and being here. I commend everybody for sticking around and trying to learn more about science, if that's what interests you, if that's what gets you going, kind of like me. Thank you very much for watching, and with that being said, don't remember to hit like, subscribe, and share. And as far as gaming content, I haven't given up on that yet. I'm still going to try to get up there with the gaming content, but you'll see much more scientific videos on my part. Thank you very much for watching. Have a great day, and I will see you people next time.